So because we're running the STS paid gels, all with each module, you will get, again, practice with it each time, and then you're going to be able to do it by yourself. Same thing with the transfers. The first time I'll set up the transfers and demonstrate them for you. The second module, when you come in, I will help you set them up, and I will have some extra for you to practice around with as well. The third time, you're going to be doing it all by yourself. So that's the way they'll go. But I highly encourage you to watch her do the techniques for each session. She, like I said, literally goes through the protocol step by step by step. And then you will notice that there will be some extra, learning. So this is, the quantitation portion for that. So first she has the running, preparing your dilutions, and then she has how to run the Bradford assay. So she'll have both of those parts in here, and then there'll be some, other videos that you can watch to kind of get the background information. All of these are important in their own way to help you learn the technique, what the system is for, what each chemical is used for, and also how to troubleshoot any issues that you have with your system. Okay. Then I have posted some practice data for you to use, since we do not have the data from your own lab because you are not performing it yourself. Actually, Tuesday lab ran into Bradford dye being messed up, even though it was a brand new bottle, but maybe it had been ordered a long time ago. It was no longer good. So they didn't get good data either, except for a couple of groups that used new and a different version of it. So they also got the same practice data. So what I would like you to do at this time is to download that practice data file. And then you're also going to download the protocol, because you need the information from there for the standard curve. So go ahead at this time and do that. I'm gonna bring this down. No, not bring this down yet. Is everyone good? Everyone got so far? So when you open the standard curve practice data, yeah, I know you can't open it. Let me change the name for this one. Actually, let me give you this one. This will save you a step. Give me 1 second, guys. Because instead of you getting that info from the protocol, we can just get it from here. So go ahead and download this one. And once I make it available, this is better one. It has the standard curve information with already inside it. Okay. So once you do that, it's going to look like this when you open it. And if I were to open up the protocol, just so you know what we would have done, this is what we would have done in the class. We would have prepared a set of dilutions with the BSA, which is our serum albumin, right? And we would have used it to run a Kumasi Blue Bradford assay. So we were going to create 20 microliters of each dilution. We were going to mix them up, And then we were going to take 5 microliters of each of them into our wells. And again, each person on the station would have made their own. So you would have had 2 sets of your standard curve. We were then going to add 250 microliters of the Bradford Kumasi binding reagent. And we want to make sure that there are no bubbles. One way to prevent that is by not going all the way down to the second stop and only going to the first stop when you add the Bradford. That prevents you from introducing those air and the bubbles inside it. And then we use the plate reader to read the absorbance at 595 nanometer, which is the point at which that blue dye can be read. And then to create our standard curve, we are going to be using those absorbances and the concentration of the protein present in each of the dilutions. We're going to subtract the blank, which has no BSA, from all the samples. And then because we made duplicate samples, we are going to get the average of those two and use a scatter plot to create the standard curve. We're not going to plot the blank, and we're not going to plot any unknown proteins because we were going to use unknown proteins as well. This time, unlike before, we're not going to just make a linear trend function. We are going to actually see which trend follows the best with our samples. So we're going to do linear, logarithmic, and polynomial regression equations. Okay, and so we'll do that in a second, step by step. I want to make sure that everyone is clear on the standard curve and the protein quantitation methods first. So let me just make sure of that first. Let me download this. Oh, I guess it works. Okay, you guys are good? Yes, no, maybe? I can't hear you.
I'd say so.
You say so? Okay.
Yeah.
Perfect. So just as a reminder, this is something we've been working with in GB1, and I'm sure in other classes too, and definitely in MoCell. We made a methylene blue standard curve. We made protein quantitation standard curve. So we've done standard curves a lot. But just to remind you that it is a type of a graph that is used in quantitative research. Anytime you're trying to quantitate protein or nucleic acid or some activity, we usually have a standard curve to go with it so that we can do so accurately. And all it does is it represents the relationship between our two, our independent and our dependent variable in some way so that we can utilize it for our needs. To create that relationship, to visualize that relationship, we have to have some type of a known sample to start with. And not only do we need one known sample, we need a range of known samples. So we need multiple samples at different concentrations or different activity levels. And then we measure those first and plot them and create our standard curve utilizing those. Now, in a typical setting, we like to create a linear trend function in our standard curve by taking just the linear portion of the graph because it makes our life easier, simply, right? If we have a simple y equals mx plus b equation, we can easily utilize it to measure our unknowns by a very simple calculation. If we have to use a logarithmic or polynomial equation, obviously, then you have to do quadratics and you have to do more complicated equations. And so, we always like a linear trend function if we can have that. Sometimes it's just not possible though. So be mindful of that, your samples or your particular assay that you're using to quantitate your proteins may not be very linear, and you may be stuck using one of the other equations. Maybe not always, but sometimes. So how are we going to use the standard curve in today's lab? Is that we are going to be using it to determine the concentration of an unknown sample. Usually it's going to be our experimental protein. Today, it's just an unknown protein that is given to you as our unknown. And our known is going to be the bovine serum albumin, BSA. It is just a standard protein. It's a purified albumin protein from serum. And we know the starting concentration of it. So we can create several dilutions from it, and we can create those known concentrations so that we can look at the relationship between those concentrations and absorbance, and then utilize that as our standard curve. Now, what happens if we don't have any bovine serum albumin available? What else can we use? And this is something that I may have asked you before as well in a different class. The
Egg
answer
yolks,
may be
right?
the same or different. Huh?
Egg yolks, right? Or yeah, egg yolks.
Egg yolks?
Yeah, wasn't there something about
Why?
egg yolks?
Why can egg yolks have a lot of fat in them? So it's not like it's just protein.
But weren't there specific growth factors also present in egg yolks that were also like yeah, I don't know, relevant here?
Oh, so you know what you're doing right now?
Yeah.
You are confusing two different things. You're confusing FPS, which we use, the serum that we use in cell culture to help the cells grow, because for those, eggs were used in the past as well.
Okay.
Egg yolks for those to provide those same growth serums. But that's
Okay.
not what we're talking about. We're talking about creating a standard curve for a protein quantitation assay. So in my case, or in your case, you will be using a pure bovine serum albumin, pure protein sample with one single protein. And we are going to be using that to create our dilutions of known to make our standard curve. If we don't have that available, what else can we use? What is important for that standard curve to have? So like I said, egg yolk won't be good because it also has fats in it, right? It has a lot of fat in it. It has some protein, but it has a lot of fat. What do we want for that standard curve sample for our known?
Could we use does it have to have albumin in it? Whatever we
No,
use? Oh, okay.
absolutely not.
Then we could use like protein powder, right? Like
Yeah,
pure
you can
protein
use
powder.
any protein. Yep. You can use any pure protein. Could be whey protein from GNC for all I care, as long as it's just pure protein. Could be any other, you get protein samples from commercially available for almost every protein nowadays, any protein that is known to be used as standard curve for other things. You can use integrin, galectin, any protein, doesn't matter what it is, just as long as it's a pure protein and you know the starting concentration. Or it could be a lyophilized protein powder and you can create your own starting concentration to work with. Okay? So that's what's important to know about that our known doesn't have to be BSA. It just has to be pure protein. That's all. And once we have the pure protein, we can create our dilutions from it and we can go ahead and utilize it. Now, to quantitate the proteins, there are two different ways to quantitate it. One is to use the amino acid absorbance wavelength. Does anyone remember what that is? So what wavelength is absorbed the most when there are amino acids present to detect amino acids?
It's in the 200s, right?
It is in 200s. You're right.
I'm thinking like like 280 or 295, one of those.
280. Perfect. It is 280 nanometer. So at 280 nanometer, you can detect amino acid content in your sample, whatever. It could be a complex sample with lipids in it, or it could be just pure protein sample. But yes, you can utilize that information from that. Now, the issue with that is that it's a very small wavelength, right? And so that means that our plate reader has to be able to detect it, which not most plate readers can do many times. And they'll be more expensive and not available worldwide to all the researchers. And also that means that we'll have to use UV compatible plates so they can be utilized for this, because that wavelength can't cross over in a simple plastic plate. Well, that obviously, again, and those plates are a little bit more expensive. So that makes it less available to people. So the alternative to that is to use a colorimetric assay that produces a color change that brings these wavelengths up to a more, kind of generally available setting that can be utilized with a simple plastic well plate or a plastic tube or something simple and worked with. There are three different different assays that have been used in the past or historically. These are colorimetric assays that produce a color change that can be detected with spectrophotometry. And in all cases, the more absorption you get, the more amount of protein you have. So generally speaking, that's the relationship. Biuret and Lowry methods, which were the first to be developed and utilized, both are based on binding of copper ions with the N-terminal of your peptides under alkaline conditions. In one case, you get a bluish color, which is the 550 nanometer wavelength. In the other one, you get more higher color change. So it's, or higher wavelength. So it's a more purplish 750 nanometer color change. Now, with Biuret assay especially, one, it requires you to heat the samples up, boil the samples up, then do something. So it's like a little bit more cumbersome. And many times it ends up with this white precipitate as well. That's going to affect your spectrophotometry results. So it's not going to work very well. Lowry method can take up to 2 hours to perform, but it's super linear, very quantitative. It's great, but who's got 2 hours to do this? Sometimes you are in a hurry or you have some protein that is super, super sensitive and can degrade very quickly, and you want to run an activity assay. You can't wait around for 2 hours. So nowadays, there is an upgraded version of these methods that is called the BCA assay that works similarly with copper ions binding to the N-terminal, but under more acidic conditions. And that's something that actually most researchers use in their lab. It only takes 15 minutes. You just incubate the proteins. Once you add the dye, you incubate them at 37 for 15 minutes and you're ready to read them. And that's great. And again, because it's a little bit slower process, it's more linear for wider range. But if you want something quick and dirty, kind of, just you want to just get it done and move on, there is another option available. It's called Kumasi blue dye binding assay, or called the Bradford method. It binds to proteins under acidic conditions and results in a change from like this brown reddish color to a blue color. And the more blue you have, the more protein you have. And this change is measured at 595 nanometer associated with that blue dye. Now, the issue with this one is that the Bradford dye, the Kumasi dye, is not very stable, and it can degrade. It's light sensitive as well, which is what we had the problem with. Usually it comes in brown bottles so that it kind of protects it from that light and is more stable. The bottles we had were clear, and I think that's what caused it, that maybe they had been there for a year or so and they were clear bottles and it degraded the dye. And so it wasn't working very well. Another issue with that is that because it is such a quick assay, as soon as you add it, you see the color change. There's like no incubation time needed, which means it's going to saturate very quickly. So if you have samples that have really high amount of protein present, it's going to saturate and be outside the linear range, and you won't be able to quantitate them very accurately at all. So you have to usually end up having to make dilutions of your proteins to make sure that your unknowns are within that range where it can quantitate them accurately. Also, it can interfere with some of the lysis buffers. So it's non-compatible with them or incompatible with them. I don't think the non-compatible is a word, sorry. So for those lysis buffers, it's not going to let you have a quantitation because it's incompatible in those conditions. So just to tell you a little bit more about that, the Bradford assay is, here is the Kumasi Brilliant Blue dye. It has several charged areas within this multiple ringed structure. And so both the structure of it, having all those rings and all those turns, helps to bind to the proteins. And also these charged areas, these negative and positively charged areas are going to bind to those charged amino acids. So charged R chains on your peptide chain, polypeptide chain. It's not always blue, obviously. It's more of this brownish black, brownish red color. It is in its normal charge cationic change when it is in the acidic pH. When it binds to proteins, it stabilizes it in its anionic or negatively charged blue form, and that's the color that you see. So it's going to have a bias, obviously, to your charged amino acids, and specifically to your negative groups. Its negative groups are going to bind to those positively charged parts of the protein. So those positively charged R groups, arginine, lysine, histidine, those are the ones that it's going to bind to most. Also, another way that it's going to bind is, like I said, with its ring interactions, because it has all these rings and turns, it's going to kind of loop around the ring amino acids. So again, tryptophan, tyrosine, and phenylalanine is going to have higher binding too. So it's going to be binding more to those than others, which is fine. It's most polypeptide chains will have some of this, so you will get those represented within your sample. Okay, so to create your standard curve, we are going to be using the BSA. Like I said, that's the first part, and you are going to be using a range of different dilutions. You want to calculate the protein concentration of them. So this one shows you 9. The protocol actually had 11. And so we actually used all 11, and that's what I've given you in the new one is right here. It has all of them. The starting concentration, tube 11, is your fully concentrated, just like stock BSA that I give you in a tube to use. It's 2 milligrams per milliliter. You want to calculate these other concentrations. How would you do that? So how do you calculate these concentrations over here? Obviously, this first one, what is this one? What should I write over here for the very first tube? That one has 20 microliters of milky water and no BS. 0, exactly. The first one is a 0. Why is it there? Why do I care for that? Why is that important?
Control.
For what?
Just water.
Just water. Yeah, so the reason for that control for water is that everything has a background noise. So once I add the Bradford agent to it, I want to make sure that that water was not contaminated with protein and doesn't give any blue in it, right? But also whatever absorbance I get from that sample is my background noise from the assay in my plate reader. And I need to subtract that from all the other absorbances before I start working with my samples. Okay, so that's the one that I'm going to subtract from everything else to get my blank corrected absorbances. And then the rest of them are going to be calculated using my favorite formula, right? So this just tells you the activity. But we're going to just work with that. I'll explain it to you as we go. So we want to calculate the rest of them, right? Using my favorite formula, which is C1 V1 equals C2 V2. How do we utilize it? So C1 V1 equals C2 V2. What's C1?
The original concentration.
The original concentration, which is what? So C1 equals original concentration, which is what? I told you the original concentration of our BSA solution.
2.
Yeah, 2 mg per ml. V1 What's going to be the V1? So it's the starting volume. What are you going to use here?
20 microliters.
No.
Ah.
0.
So we're talking about the original concentration of BSA. So for V1, we are going to use someone said 0. No, no, no. Well, let's look at tube 2. For tube 2, what will I use for V1? You guys can see the numbers, right? No, that's the water. We are trying to find the amount of protein present in them. Yes, it's going to be the starting amount of protein. So starting volume or volume of BSA used for that particular tube. So in 2, we will use 2. For tube 3, we will use 4, 6, 8, and so on. Okay. Then for C2, what is that? Kaylin, is that 2 for this question or the past? Oh, legs are killing me. Sorry. Oh, okay, that's fine. Yes, 2 was for the last one. So for your ending concentration, what is that? That's what we're trying to find. Exactly, that's your X. And then that's the final concentration of your sample. And then V2 is going to equal what?
20.
20. That's our final volume of the dilutions we are creating. So that's going to be 20 microliters. Perfect. So use this formula and tell me the concentrations, or, let's use this formula. I'll give you guys 5 minutes and you let me know what your concentrations come out to be. Okay. And then we'll fill out this chart so you have this information available. I'm gonna pause the recording for a minute. Anyone finished doing tube 2? Once you do 1, it should be easy. You can even use Excel to do it. Just create a formula for it. Right? We could solve for X like this. Let's actually make a formula together. That might be more fun. So what should I write after equal sign? Equals.
Or divided by 20.
Not quite, because we have to take into the account the original concentration. So it's going to be 2, which is the original concentration first, right? For volume of BSA, I want to use whatever's in this row or column. So I can select for that. And I want to put them both in the parentheses so it knows that's all the top portion. And then for my denominator, what should I write? Divided by
20.
20. Okay. Where did I go wrong? Oh, I didn't put this correctly. I need one more. There. Yeah. Come on, tell me what you are saying. I don't see you. Where am I? So 2 multiplied by T4 divided. Hey, stop. You're not showing me your word, so I can correct you. Yes.
Professor,
Yes.
sometimes it wants you to put an asterisk when you multiply.
Oh, is that its new thing after it's
Yeah.
upgraded it?
think
Okay,
so.
fine. That's what it's probably saying, but it's having a meltdown because it's unhappy with my life, with my choices. Okay, well, that's the way to go, Excel, making things more complicated after updates. It's also made it, it recently updated and now it's like all black. I hate it. I wish it was white again. But that's okay. I'll deal with it. There we go. So did everyone get 0.2 for this one? For the first one?
Yes.
Okay. Now I want you to be able to do it with a pen and pencil too. Is it? I don't know. It's making me more depressed than anything. Maybe I just have to get used to the dark mode. Now, the fun thing about this is that because I made a formula, you should be able to just bring it down and it will give you all the other numbers. Okay? You can go even down and it will give you the 2 as well. And you can make sure your formula was correct by double checking it that way, that your last tube should give you 2.
I have a question.
Yes, dear?
So I'm trying to do it in the Excel, but whenever I click enter, it keeps showing up as a date instead
As a
of
date?
yes, it keeps saying 01 January.
How
or
fun.
something.
Okay,
Yeah.
I'll stop my share. Go ahead and share your screen and let's see if I can help you out, okay? Go ahead, share your screen.
Are you able
Okay,
to
so
see
I see it. I wouldn't do it here. Let's just try doing it where that column is itself.
here?
No, green area where it says BSA concentration milligram per mil, because that's where you're trying to do it right there. So let's write it there. Equal sign and parentheses, another parentheses. No, the other way, going the same way. Don't close it yet. Open, open, yes. And then the first one is going to be C1, so it's 2. Close parentheses, asterisk, open parentheses. Click on the column next to it, the 0. Yes. Close parentheses, another close parentheses, divided by 20, because that's the total volume. Equal. There.
Oh,
Now go there, top again, and drag and drop all the way. Yeah.
okay.
I would even drag and drop to the 2 and make sure you get 2. Because that's always a good internal check. There you go. Go to that.
Thank you.
That just, yeah, I don't know what happened there, what its issue with life was. Okay, so we got that so far. Everyone good? So that's going to be our X or Y. Which axis is that going to go on? The concentration of BSA. Is that our independent variable or our dependent variable?
All right. Independent variable, so it'll go on the
Exactly.
X.
Perfect. So independent variables go on the X. That's our X-axis for our standard curve. Now, the next thing we're going to do is we're going to take our data for the actual absorbances. These are the absorbances that we have gotten from the 2 students, so-called, right? So this is student 1 on the station, and this is student 2 on the station. That's their numbers. So you are going to select these, and you can notice that the numbers are a little bit different, right? They're not like they're perfectly aligned. That's fine. So copy, Control C, and then you can do paste special and do transpose and it will get the numbers in the column. Isn't that nice? Okay. And so this is absorbance 595. And to tell you even more about them, these are raw absorbance 595. Student 1, student 2, and this is, I'm gonna do merge. The more information you provide to yourself or me, the better it is. Raw absorbance at 595 because it's not blank corrected yet. Okay. And now, Control C, we are gonna have the blank corrected absorbances. And so I'm gonna make them different colors so we see it and clear this. Okay, so let's create a formula to get a blank corrected absorbance. What should we write? What do I want to subtract from all my values for student 1? I lost my chat again. The chat likes to run away. Yes, and for student 2? Yes, so let's write equal sign, and I'm gonna make a formula so that it will, I make one formula and I can just drag it down and over and it will do it. So I will say equals, let's select this first one, minus what? Should I do, I just want to write one formula that's gonna cover both. So if I do J33, what do I need to put to make sure that it takes J33 for all of student 1 and K33 for all of student 2? Dollar sign where? Do I want to put a dollar sign for both J and 33? Just J or just 33? What do I want to keep constant?
Just J.
Just J? Are you sure?
Yeah, because it'll lock the whole column of J, right?
Not quite. So it will always keep J constant, but it will change when we go to the next one. It will still be J. So for student 2, it will still take J. Is that what you would like?
No, so in front of the J and the number.
Then it will always take J33. Both J and 33. So it will always take that one single well. One last time.
So just the 33.
Just the 33. Yes. So if you put it in front of J, J will always be constant, but it's going to change the row as you go. So everyone's going to be 0 on student 1. And it's still going to be a weird number for student 2. If you put it in front of both J and 33, it's going to always take 0.354. Always take what's in J33, not 0.287 when you shift to student 2. By putting it just in front of 33, you're making sure that the row is constant, but for each column, you're taking its own blank. Okay? So let's see if that actually is true. Right? Now you have 0, 0. And now I can drag it down and I have my blank corrected values. Sounds good? So finally, we are ready to combine the data the way we want for our actual standard curve. So I'm going to make a new little chart here. So I'm going to have the BSA concentration in milligrams per milliliter against absorbance 595 nanometer. And this is going to be average absorbance, so I should probably write average absorbance at 595. For this one, I'm going to write whatever is up here. And for this one, I'm going to average equal sign average. Oh, not blank average. Where is the formula for just average? There. Average, and I'm going to average whatever is over here. Now, notice that I did not start with 0, 0 because we are not going to plot the 0, 0 point. The blank is just going to be out right there. And now I can go ahead and drag and drop for the next 10 rows, which this was completely by chance. I did not actually notice, but it worked out. Cool. Everyone got so far? So finally, we are ready to plot it and get our standard curves. What's the formula for BSA? I literally just wrote equal sign and I clicked on 0.2, whatever is in that well that we calculated, or like that column that we calculated, that particular slot. So I said E34, right? Because that's where it was. And then when I dragged and dropped, it automatically took whatever was in the next well, next. Cool. Okay, so the rest of it, you guys should be able to do pretty much. I'll give you a little bit. So then you're gonna select these two. Once you have them, you are gonna go to insert, charts, scatter plot. Wow, that's surprisingly linear for whatever reason. And then you're gonna format it according to how we have learned to format for many, many, many years at this point. By going to the chart elements, I'm gonna add chart axis titles. You can keep a chart title for this time if you want, that's fine. We're gonna remove the grid lines. You're gonna add trend lines. Now you're gonna have the same graph 3 times with each one with its own trend line, because we're gonna do all types of trend lines. This first one is a linear trend line. Then you're gonna do one with exponential and one or logarithmic, and then one with polynomial. Okay. So here, what you want to make sure is that for each one of those, you display the equation and display the R square. And for your chart titles, you can call them by that. So it could be standard curve, linear trend function, and then, right? And then you can write polynomial and logarithmic for the other two. That way it's very clear to you and to everyone else what's being shown in each one. The other thing you would want to do, just to make it better, is again format your chart area to have no line for borders. Same thing inside, no line for borders. You want to make sure your axis lines, however, are visible. Solid line in black. Same thing for your other axis, solid line in black. Then you also want to make sure that your numbers are, not so crazy. Go back to access option. This is fine. So let's go. I'm gonna go here and go to format axis. And I'm gonna make the axis to 1. And I'm gonna do major units a little bit further apart to make it less busy. So 0.2, right? And then 0.2. And then I want to make sure that the numbers all look the same. So I will do number and just do 1 decimal point. In tables, we don't want to make them super busy. I said 1. Do you not know 1? There, perfect. And then you can change the size of each one of these to make it better visible. This is gonna be your BSA in milligrams per mil. This is gonna be your average absorbance. 595 nanometer, right? This over bold. Bold. It's clear what's there. Make these at least 10, if not 12. There we go. And that's a properly formatted graph for you. Now, you could just duplicate it, frankly, right? And for this one, you can go to your trend line and more options, and you could do logarithmic. The rest of the stuff remains the same, and you say display equation, right? And delete. Why isn't it deleting that? Normally I can delete the linear. I just, no. I only want logarithmic in that one. I do not want anything else. I don't know why it's not doing that. I will have to find out what this new system has done. So if that's the case, what you can do is you could remove the trend line from the first one before you copy it 3 times first, and then make your different equations for each one to make it easier. Control T. You can have your 3 side by side. Apparently because of one or up, down. And then for each one, you can make your different trend line. Linear options. Display R square. Just make sure those are visible. The second one will be our logarithmic. And we want to go to more options. And then polynomial. It's called 2-order polynomial in here. Right there. That's it. There's a display R square. And then you examine them and you see which one is the most appropriate to use. How would you do that, by the way? How would you know which one is the most appropriate for the data that you have been provided?
The R squared value is closer to 1.
Exactly. The one with the closest R squared to 1 is the one that is the most appropriate. And in your case, which one is it in this particular data set? Thankfully, it's not logarithmic for sure. My goodness. That would have been sad. The polynomial, exactly. So you would want to use that one to do your unknowns, okay? So that one does require you to use the quadratic equation. Now for your unknowns, you have your unknowns right here. The first number is your 0, so it is no protein, no sample present. This is your blank. For those two, again, there's two numbers because student 1 and student 2. The second one is a 1/2 dilution, okay? 1 to 2 dilution. No, don't be like that. 1/2 dilution, of course. I never know what it's doing. And this one is your undiluted. So this is 1, pure unknown by itself. Okay, so you're going to be correcting them, blank correcting them, and then you will be utilizing the numbers to figure out your concentration using the appropriate equation. Now, does it matter which one you use, undiluted versus the 1/2 dilution? Which one, yeah, does it matter which one you use, first of all? You guys there? How do you know which one should be used? Is there a design to the madness? I'll keep it here. So in an ideal world, well, there are two things that matter when you're doing your unknowns. One is the accuracy of your unknown dilutions. So if you made your dilutions perfectly so that your 1/2 is exactly half of your undiluted, then it doesn't matter which one you use, you will get the same answer at the end of the day using the appropriate equation. That's step 1. These seem to be reasonably made within, 10% or so error should be fine. The second part is which one fits your standard curve. A standard curve is more appropriate and most quantitative if used for unknowns that fall within its range. So they should be between 0.2 to 2 milligrams. If you have a sample that's above 2, it's not going to be as accurate in determining it with the standard curve because it's outside the range of your standard curve. Now, obviously, we don't know what our unknown's concentration is. That's what we're trying to figure out. But we do know the absorbance. And we can figure out if our blank corrected absorbances of our unknown fall within the range of our standard curve, right? So let's look at the range of our standard curve using the average absorbances. Looking at these numbers, what is the range of my standard curve? What is it most accurate for as far as the absorbance is concerned? Range of standard curve.
From around for absorbance 0.09 to 0.85.
Exactly. So our curve is accurate for absorbances 0.0, in my case 0.093 to 0.853. Cool. That's the range of my standard curve. So once you blank correct your unknowns, because see, these unknowns have higher blank value for whatever reason as well. So once you blank correct them, you want to see which one of your absorbances fall within the range of the standard curve. Again, you're going to do the average. I'm not going to do this for you because I want you to do this step yourself. You're going to blank correct them just like we did before. You're going to average them just like we did here. And then you're going to see which one half or the fully undiluted falls within the range of your standard curve. You're going to utilize that absorbance, just that one, and you are going to calculate using the equation that is most appropriate, the concentration. Where will the absorbance go? Will it go for Y or X? Where would you put it?
Why?
Y, exactly. And you're going to solve for X. Now, if you're using the quadratic formula, remember you get two answers and you'd see which one makes sense, right? So one of them will make more sense than other for sure, and that's the correct answer. Everyone good? Yes? No?
Yes.
Perfect. So that's our lesson for today. I will have this recording available online as well for you to go back and look at. And you will be figuring out those unknowns yourself and bringing them to class. And we will discuss it in the beginning of class. Can I scroll up to the range? You can look at your own range, Yashvi.
So
The
is there
range
anything?
is
Oh,
the
sorry.
range is going to be the absorbance of your lowest concentration of protein all the way to the highest absorbance. That's the range. Yes, Jason?
Is there anything you want us to submit from this?
No, you're not submitting anything. This is practice. So we are giving you kind of a freebie in this one.
Gotcha.
This is just to make sure everyone is up to track and knows how to do this. And I'll just be looking at them in class and making sure that you've done them.
Sounds good.
Okay, perfect. So I'll see you not next week, but the following week in lab, and be prepared to do a 4-hour fun lab.
Yeah.
Just tested it out. It looks so good. I love affinity chromatography. So it's fun. Okay. I look forward to seeing you then. Take care, guys.
Thank
Bye-bye.
Have a good one,
you.
Dr. Adam.
Take care. Bye-bye.
Bye-bye.